Hello everybody. Today we're going to talk about the microbial ecosystems of hydrothermal vents. And the main point I want you to get from this video is that the discovery of hydrothermal vents was a major geological discovery, and it was also a major biological discovery. So let me explain what I mean by that. So first of all, hydrothermal vents are found primarily on mid-ocean ridges, the places in the ocean where the oceanic plates are spreading apart. Here is the Mid-Atlantic Ridge, the part of the mid-ocean ridge in the middle Atlantic Ocean. And in the middle of the Atlantic Ocean is this gigantic mountain chain, one of the largest mountain chains on Earth. And these mountains are formed where the ocean crust is literally being split apart. So you probably know that North America and South America are splitting apart from Europe and Africa, literally being stretched apart like this, and that is happening because the ocean crust is being split right here. So the Mid-Atlantic Ridge is being formed by the splitting of the oceanic crust, which then moves the continents. So the discovery of hydrothermal vents was came about by geologists trying to figure out where was all the heat escaping as a result of continental drift. So the idea of continental drift had been around for a while, and it was mostly accepted by geologists through most of the 20th century. But there were a few holdouts because there was this problem of, okay, if these continental and oceanic plates are moving around the globe, then that means that there must be a lot of energy associated with those plates moving around, and that means there must be a lot of heat coming out of the Earth somewhere as a result of all this movement. And geologists had predicted that that heat must be coming out in these places where the plates were splitting and spreading at the origin of the spreading. But nobody had been to the seafloor before with a camera or with human eyes. And so we really didn't know what was down there until the late 1970s and 1980s was really the first time that anybody had ever gone to the seafloor with a camera and human eyes. And that is often a surprise to people that it took us that long to make these discoveries. So anyway, geologists predicted that there must be places where heat was coming out of the seafloor, and we now know that the way this works is magma from the Earth's mantle rises in the weak spots created from the oceanic crust splitting apart. So when the seafloor splits at these mid-ocean ridges, also known as spreading centers, that creates a weak spot, and that allows heat to come up into the seafloor. The heat heats up water that is circulating through the oceanic crust. The hot water rises and pours out of what we now know are vents here. But the first time that anybody had ever seen these vents was actually in 1979. Was the first time we saw hydrothermal chimneys. In 1977 was the first time that anyone had seen kind of the consequences of that hydrothermal circulation. So I'm going to tell you about those discoveries. So here's a photo from a camera that was being towed behind a ship in the Pacific Ocean near the Galapagos Islands in 1976. So this was not a submarine. There weren't people down there. This was just a camera that was thrown overboard and connected to the ship with a cable. And the geologists had predicted that a lot of this heat coming out of the Earth must be coming out around the Galapagos vents the Galapagos Islands for geological reasons that we won't go into in this class. But anyway, that's why they were taking pictures of the seafloor at that spot in the ocean, because they thought there should be some heat coming out. They weren't expecting anything else except maybe looking for some tiny volcanoes or some funny rocks that looked like they might be associated with heat. And so in May 1976, they took this photo where they had some funny white stains on the rocks on the seafloor, which we now know are hydrothermal precipitates. But when they first saw this photo, their first thought actually was that this was just a result of a bad photo development, because these were the days where they were developing their own film negatives by hand with chemicals on the ship. So they thought maybe this was just an artifact of the photo processing. They saw these clams that they could tell were clams, but they looked kind of dead, and some clams that they could tell were buried with soot. So the soot was really exciting because that was some confirmation that there was some kind of volcanic activity going on here. And they also found an empty beer can. And so they didn't really expect to see a whole lot of animals here. There was no reason to expect a lot of animals. So one interpretation was they had come across a dump site that a party boat had gone by. This was, near the Galapagos Islands in South America. There was some tourism there. So their first thought was there was a party boat. They were eating a lot of clams. They dumped them overboard, as well as their beer cans. And that was at least one possibility. With more studying of these photos, biologists realized that these clams probably were real. They really were living there. And it was 1977, so about a year later, they went back with Alvin, our one and only manned submersible for scientific research in the US, and got some much better photos of live clams living on the seafloor in these big clumps. And when they brought the clams up, first of all, they're huge. These are gigantic clams. And inside, the clams are red, are blood red, because in fact their blood is red, which is unusual for a marine organism, as we'll talk about in a little bit. And when they first came across these big clumps of animals, there's this famous conversation between the scientists inside the submarine, where the geologist, Corliss, says, "Isn't the deep ocean supposed to be like a desert?" And when the biologist answered yes, Corliss replied, "Well, there's all these animals down here." So this was the first discovery of big clumps of animals near or on the seafloor in general. And it turned out it was because they were near hydrothermal vents. This was in 1977. In addition to the giant bloody clams, there were also these weird animals that we now know are called tube worms. Tube worms because they live inside these tubes that they make, and worms because they are in fact annelid worms that live inside the tubes. These can be up to 6 feet tall, so as tall as a human, and they have these funny red lips that are technically called plumes that we'll talk more about in a second. First, let's just focus on this initial discovery. So that Alvin dive was 1977. The paper in Science came out early 1979. Submarine thermal springs on the Galapagos Rift. So this one paper is reporting a big geological discovery that they had finally found the hot springs that are releasing all of this heat associated with continental drift. And if anybody had still been skeptical about the idea of continental drift at this point, this final nail in the coffin, this final geophysical confirmation of all of this heat being released was the proof that this is really how continental drift works. So there's that big geological discovery, the final proof of continental drift and how plate tectonics works and where the heat is being released. But at the same time, this is also a huge biological discovery. So in the summary of the science paper, the last couple sentences are the vent areas are populated by animal communities. They appear to utilize chemosynthesis by sulfur oxidizing bacteria to drive their entire energy supply from reactions between the seawater and the rocks at high temperatures rather than photosynthesis. So instead of an ecosystem being driven by sunlight and photosynthesis, instead we have these giant animal communities that are probably being supported by chemosynthesis instead of photosynthesis. And we're going to talk a lot more about that in a second, but I think it was interesting to point out that they had already realized the significance of this biological discovery at the very beginning. So a little more detail, some actual paragraphs from this paper. This is one of the key paragraphs. Perhaps the most important question about these animal communities concerns their source of food. Particular features of the Galapagos hot spring environment, such as the depth and abundance and size of the animals, remember these animals are huge, suggest that the supportive organic nutrients could not be substantially derived from primary productivity in the surface waters. In other words, you couldn't get gigantic animals at high density just feeding on the little bits that were, sinking down from the surface. Moreover, because of the high concentrations of hydrogen sulfide in these waters, so they had already noticed that when they brought these clams and worms up to the surface, they stunk like rotten eggs because they were totally soaked in hydrogen sulfide. So a significant proportion of the organic carbon utilized within these hot spring regions could be produced by chemolithotrophic sulfur-oxidizing bacteria. In our class, we've been calling them chemoautotrophic, chemolithotrophic, or chemolithoautotrophic. They're all basically the same words, slightly different words for the same idea. So indeed, they found huge concentrations of bacteria in these waters, and these counts, by the way, were done by my PhD advisor, obviously long before I was a student. And then the significance of this was already realized by my PhD advisor and the other authors in this paper with this final sentence, this flux of bacteria from the vents. So they're finding huge numbers of bacteria pouring out of the vents. This flux of bacteria from the vents must be supported by the productivity of a large population of bacteria living within the rock mass, lining the walls of fissures through which the hydrogen sulfide-laden fluids ascend. So they're seeing huge populations of bacteria fluxing out of the vents, which means that those bacteria must be coming from somewhere, which must be a large reservoir of bacteria living underneath the vents, literally in the rocks and the cracks and fissures in the rocks where the water is flowing and where the hydrogen sulfide, the energy source, is coming from. So this initial discovery is a window into a huge subsurface habitat that nobody had known had existed before. All right, so that was the big discovery. Now let's talk a little bit more about what we know now about these animals, because they're pretty awesome. So again, here are those giant clams. Not only are they large, but they grow very quickly, several centimeters per year. Here are the tube worms. They can grow almost a meter per year, which is pretty impressive. They're really fast growing. In this photo, you can also see how the worms themselves are habitats for many other animals. So you can see these crabs crawling along the worms, and I think that they will eat the worms if they can, but mostly they're eating much smaller animals who are living on the tubes of the tube worms. We'll see that in a video in a second. And it was noticed that these tube worms are living very close to the source of the fluid. So here's a nice photo taken later on, this was in the mid-90s, of super, super hot water coming out of a hydrothermal chimney here. And you can see that these tube worms are living very close to the source of the super hot fluid coming out of the chimney, but not right next to it. In fact, right next to this black smoke, which indicates the hottest water, you see actually a bunch of dead animals that were probably living here very happily just when the plumbing of the hydrothermal system shifted and suddenly a fire hose of 400 degrees Celsius water came shooting out of here and fried everything that was living near it. So this is why there's some fried animals here, and then in a ring around the fried dead animals are nice, happy, healthy animals living in the sweet spot of being close to the source of their energy. This is where the hydrogen sulfide is coming out of, but not so close that they're getting too hot and being toasted. Here is another photo of one of these giant clams, and you can see how when the scientists dissected those clams, it's just very bloody, and they quickly discovered that it was blood red, in fact, because they have iron hemoglobin, just like we do, which is like us, but completely different from all other marine invertebrates. So a normal clam does not have red blood. It has a greenish-bluish blood because the hemes in its blood are made out of copper. Normally, for marine invertebrates, these are unusual, and that these giant clams actually have iron hemoglobin just like us. I'll explain why in a second. Here is a diagram of a tube worm to explain a little bit more about the weird anatomy of these worms. So again, the worms live inside these tubes that they make for themselves. They happen to be annelids, so they are in the same group as earthworms. You can see in this diagram, at least, how they are segmented in this way. They have no mouth and no anus. So the only way that they can bring in nutrients is through these red plumes that look like lips. But it's not a mouth in that the mouth does not move to capture food. Instead, the plume contains, again, red blood that captures hydrogen sulfide and brings the hydrogen sulfide through the blood into the body of the worm. Inside the body of the worm, there is no digestive system, as you would expect for a normal annelid. Instead, there's this thing called a trophosome, which is basically a giant bag of bacteria that is the digestive system of the worm. Here's a better photo of the plumes of the tube worms. This is actually a slightly different species of tube worm than we were seeing before. The plume is a little more feathery rather than those kind of lip-like features in the other species of tube worm. And here's yet another species of worm that we'll talk about in a second, which is actually a polychaete instead of an annelid. But anyway, there's a nice photo of these plumes. And so the way this works is again, the iron heme of the blood of the tube worm binds oxygen. That's what our heme also does, so in that way it is the same. But it also binds, the iron heme also binds hydrogen sulfide at the same time that it binds oxygen. So our blood also binds hydrogen sulfide. That is why hydrogen sulfide is toxic to us. So if you hang out in a hot tub or a hot spring too long, if it's a sulfur spring, if you hang out in that hot spring too long, you could actually pass out due to lack of oxygen because all of your hemoglobin is saturated with hydrogen sulfide instead of oxygen. Because our hemoglobin actually binds to hydrogen sulfide better than it does to oxygen, and that's normally not a problem because we don't normally live in very sulfuric environments. So the hemoglobin of the tube worms is adapted to bind both oxygen and hydrogen sulfide relatively similarly so that it can bind both of them and bring both of them down to the bacteria in its modified gut. So then the bacteria use hydrogen sulfide as an electron donor. They use oxygen as an electron acceptor for a chemoautotrophic metabolism where they use that as their energy source, and the CO2 that is also brought down to the bacteria by the worm as its energy source. It makes organic molecules with its autotrophic carbon fixation pathway, and then those organic molecules then leak out to the worm, and that is how the worm gets all of its food. So the worm eats by feeding its bacteria. Bacteria make food, and then the worm eats that food. So the worms do not forage or consume food particles in any way. The only way that they can make food is by bringing hydrogen sulfide into their body via their blood. Pretty wild. A little more detail of how this works. You don't really need to worry about these details, but in case you're curious exactly how this works, one of the main things that the bacteria or the symbiotic bacteria do is that they actually make the nucleic acids for the worm. So in addition to just making food in general that the worm can eat for getting energy, the worm actually can't even make its own DNA except with the help of its bacteria. So here's the whole pathway that occurs in the bacterial symbiont. Here's the host, and this is showing how the bacteria actually makes uracil, one of the molecules you need for making RNA, and then gives that uracil, gives those nucleotides to the worm so the worm can actually make its own DNA. So this is just to show the degree to which the worm is completely dependent on the bacteria for its metabolism. So then the host uses these organic molecules, recycles the ammonia and the carbon dioxide back to the bacteria. So this is just to summarize what we've talked about so far, and again making the analogy to photosynthesis as the primary producing process in a normal sun-powered ecosystem. Instead of photosynthesis, here we have chemosynthesis, and what we mean by chemosynthesis is that carbon fixation is happening by bacteria inside the bodies of the worms, inside the bodies of the clams. Works in a similar but slightly different way in the clams as in the worms. The bacteria are fixing the carbon dioxide into food for the animals, and doing that and powering that carbon fixation by the oxidation of hydrogen sulfide that is coming out of the deep sea vents. So it's a whole animal-rich ecosystem powered by chemoautotrophic bacteria using hydrogen sulfide to fix carbon dioxide. All right, so I've been talking long enough. I thought maybe we would reward ourselves with a cool video of these animals taken with modern camera technology rather than those fuzzy photos from the 70s and 80s. So here is a video recorded in 2011 to the same site, the Galapagos Rift, the same site where those initial discoveries were made, but with a more recent expedition. So here you see these seafloor rocks with these white staining that were seen in that original photo. We now know are mineral precipitates from the hydrothermal activity. Here you're not going to see any super, super hot water coming out of a focused fire hose-like chimney. Here the venting is more diffuse, we say, which is to say that it's just kind of coming out a little bit everywhere, making the whole place kind of like a nice lukewarm temperature instead of a single fire hose spouting super hot water. Here you can see the kind of foggy water is because it's warmer than the water around it. So this is kind of like a nice warm bath. You see the plumes of these tube worms here. You can see some shrimp-like animals crawling along the surfaces of the tubes, eating smaller critters also living on the tubes of the worms. Here you can see that the worms are often concentrated in specific spots where there's more of the hydrothermal sulfur-rich water coming out of the rocks. Here you can see how, I think if you watch this worm right here, you will see it react to a crab that was trying to eat it, and so that is a defensive behavior of the worms in response to predators. And remember that these worms can get 6 feet tall, so these are human-sized worms. So another close-up of those worm tubes. These, I believe, are probably limpets that are growing on the surfaces of the tubes. There's the shrimp again. And this video goes on for many minutes, and I will make it available to you on Canvas if you want to watch the rest of it. Okay, we've talked about those giant clams. We've talked about a couple of different species of tube worms. Here's another species of worm. This is called the Pompeii worm. It's the highest temperature animal yet discovered. It also lives in tubes, but instead of being an annelid, this Pompeii worm, the genus name is Alvinella, named after the submarine, is a polychaete instead of an annelid. It has these structures like this instead of the blood red plumes of the other tube worms. So their anterior end, can't really say mouth because they don't have a mouth, but their anterior end is bathed in this lukewarm 20 degrees Celsius water. So that's nice room temperature, nice warm bath water. But its posterior end, the rear end that is in the bottom of the tube that is attached to the hot rock, that end can be up to 70 to 80 degrees Celsius, which is super, super hot. That is not a temperature that any other animal can survive for very long. Biochemistry of the worm's enzymes indicate that it probably isn't very metabolically active above about 40 to 50 degrees Celsius, but it clearly can handle super high temperatures without dying. And as far as we know, it doesn't have any endosymbionts that are running its digestive system in the way that the tube worms do, but it does have a rich coating of bacteria on the outside of its body. So that's the difference between an endosymbiont and an episymbiont. An episymbiont is growing on the outside of the animal body. And there's a whole ecosystem of microbes growing on the outsides of the bodies, including sulfur oxidizers, sulfate reducers, nitrifiers, nitrate respirers, denitrifiers, nitrogen fixers. Many of them are very resistant to the heavy metals that are pouring out of the chimneys themselves. Another species are shrimp. So for some reason, all of the tube worms are only in the Pacific and the Indian Oceans, and there are no tube worms in the Atlantic Ocean, which is an observation of animal biogeography that, as far as I know, doesn't have a clear answer. But anyway, instead of tube worms, the dominant animal in many of the Atlantic hydrothermal systems are these shrimp. So here you can see these shrimp crawling along these rusty rocks. The rocks are rusty colored because of iron oxidation by microbes near hydrothermal chimneys. And these shrimp can often get to super, super high densities. Here's a real photo of shrimp on the seafloor. And they're all crowding around the source of warm, sulfur-rich water because they also have symbiotic bacteria that use sulfur as an energy source. So here is a microscope image of the mouth parts of the shrimp. And different kinds of bacteria are labeled in different colors, so you can see the diversity. Exactly what these bacteria are doing on the mouth parts of the shrimp is still being figured out, but it's clear that they are chemoautotrophs. They are fixing carbon dioxide, powered by sulfur, and also probably hydrogen gas as their electron donors. And so what happens is that the shrimp are crowding around the source of the sulfur-rich water coming out of the rocks because they're literally gardening the bacteria growing on their mouthparts. So they are gardening a beard of bacteria on their mouthparts, and when that beard gets lush enough, they just literally eat the bacteria that are growing on their mouthparts. And it's been shown that that is the primary and perhaps the only way that the shrimp obtain food, is by eating the carbon-fixing bacteria growing on their mouthparts. Here's another one. This is called a Yeti crab. It has these super hairy arms. It's also known as the Hasselhoff crab. And it is known that these hairs are coated with microbial biofilms. It's not exactly clear what the microbial biofilms are doing. It's, I think, a good assumption or a good prediction that it's going to be a similar story as with the beards of the shrimp, and that the Yeti crab is probably eating the biofilms that it grows on its hairy arms. But as far as I know, they haven't quite worked out the details of how this particular episymbiosis is working. It's a relatively new discovery. Here's the mussel, so similar to the clams, but these are mussels instead of clams. They are found near hydrothermal vents, usually not the super hot vents, more of the mild vents. And they're also very common near methane seeps, which is another habitat. It's not hydrothermal. Methane seeps are not necessarily, anyway, formed by hydrothermal processes, that is not by hot water, but just by deposits of methane, deposits of minerals that are trapping methane on the seafloor. So that may be a topic for another day. But anyway, these methane, these mussels not only have sulfide oxidizing endosymbionts, just like the clams and the tube worms, they also have endosymbiotic bacteria that eat the methane. So that's a reason why they're found in methane seeps. So some of their endosymbiotic bacteria are using methane as the electron donor and then pass on food to the host. In addition to having these two different endosymbionts, the mussels can also filter feed, just like any normal mussel. So they're very versatile in how they can obtain food. And I've seen recent studies that I'm not showing here where there may even be a third symbiont that uses hydrogen as its electron donor. So a third type in addition to the methane oxidizing symbionts and the sulfur oxidizing symbionts that you see here. Here's a really good paper if you want to read more about this particular topic. So finally, I just want to leave you with the idea that these are fully functioning ecosystems powered by microbial carbon fixation on the seafloor. It's not just a couple of animals here or there. These animals are not isolated. They live in very high population densities and together as part of ecosystems. So here's one of my favorite videos, even though it's not very high resolution. This is a site where there's kind of lukewarm water diffusely venting all over the place, so you don't necessarily see the water. And these mussels are completely covering all of the surface area where methane and sulfur-rich water is coming out. And then living on the shells of the mussels are limpets and other tiny animals, and then you have shrimps eating those little critters, and you have crabs eating everybody else. And it's just a very rich, very diverse ecosystem, all powered by the chemicals coming out of the hydrothermal vents. All right, that will do it. If you want to watch more videos, I have lots of them. Two of them in particular I would recommend is this interview with Bob Ballard, who was involved in those initial discoveries, where he tells Bill Nye about his personal experiences in making these discoveries. So just click on this link to watch that YouTube video. And this was a really good explanation and goes into a little bit more detail on how this metabolic symbiosis in the tube worms works. So if you want to learn more about those details, I highly recommend this video from Ed Yong. Click on this link to learn more about that. All right, that's all. Check out the assignments posted on Canvas, and I'll see you next time. Bye-bye.